Okay, here is Chapter 1, Section 3, the Methods of Science. So the main idea here is to know that biologists use specific methods when conducting research. And I think that you'll find it's not much different than your own set of problem solving that you do every day. So, if you think about it, what do you do to find answers to questions? Are your methods haphazard? Are they methodical? So in class it was brought up that if you have a question and you want an answer to it, you probably would consult Google or Siri or Alexa or something like that to find the answer. So you would do some research in order to find an answer to your question. Or if maybe you had a problem, there might be a strategic way that you would solve that problem. The example that I gave in class was, what would you do if you lost your car keys? So if you have a problem, you need your car keys, you would probably do a set of steps. You'd probably retrace your steps as the first thing. So say you know that you always come through the kitchen every time you get home, so your first thought would be to check in the kitchen, maybe on the kitchen counter, to find your car keys. So if you go to the kitchen and look on the counter and your keys are not there, then what would you do? So I bet that you would think of the next logical room that you are probably in, say the living room, because you came in through the kitchen and then you went to the living room and maybe watched some TV. So the next thing you would do is go to the living room. And if you go in the living room and you find your keys, great, your problem has been solved and you have your keys. So if you go through that type of process, that's actually going through the scientific method. We just don't really think of it in those terms. But when you're having your problem, you state your problem, I need my car keys. Your hypothesis is, and I don't like using the word educated guess, but there's got to be some thought to it. Say, like, I know it's logical that I would go to the kitchen because that's the first room I would have gone in. So I had some background knowledge there. I'd go there, but then when you go and you don't find them, you find your hypothesis was wrong, that your keys were not in the kitchen. So you have to reformulate that. Well, my next logical step would be, and my next hypothesis would be, that they're in the living room. So when you found them in the living room, you confirmed your second hypothesis, and anyway, you were able to find your keys. So I know we don't really think of it in that term, but that's really the problem solving that takes place even with the scientific method in the scientific community. So here's our vocab words. We're going to be going through those throughout the lesson. And our main objectives are going to be to know the difference between an observation and an inference, to know what the difference is between a control, an independent variable, and a dependent variable, to learn scientific methods a biologist uses for research, and then the importance of the metric system and the SI system. So scientific methods are methods that scientists use to gather data and answer questions. So it can be kind of, I know in science sometimes we look at them as like this rigid list, but it's not so rigid sometimes. Sometimes you have to go back and forth and understand it. But they all do start certain ways. So we'll go over those steps. So if I was going to put a list to it, the scientific method is really, our first one is an observation, or from that observation we ask a question. Then we formulate a hypothesis. Then we experiment to see if our hypothesis is correct. We collect data, we analyze that data, and then report any conclusions that we have. I want to say it's not always so cut and dry. Sometimes it's a process. So I like this set of memes where the scientific process is a lot of these things that happen. So in our key example, we might have had the original hypothesis that it was in the kitchen, but it wasn't. So we experiment by looking, and it wasn't in there, or we look at our results. So we can, in scientific research, if we have a hypothesis, it becomes a prediction, we experiment, and then we look at our results. So if our results is supporting that first hypothesis, then who knows, it might eventually go into theory. But if our results do not support our hypothesis, we have to reject it and then start over. So sometimes it's kind of this process going back and forth between any of these steps. So our first step, asking a question. So scientific inquiry begins with an observation, and observations are a direct method of gathering information in an orderly way. So, for example, if there was a bird that was outside that you were curious about, maybe a bird came to your window every morning, and you want to identify it. First, you're going to make observations about it, maybe how it behaves, what it eats, maybe the color or pattern on its feathers, or maybe the time of day that it comes, or even the song that it sings. So you might draw it or photograph it or make note of those different things. So again, it first starts, we have that observation and we start asking questions about it. So if you see that bird, you might want to know what that bird is. So we can furthermore find research from reliable sources. So you can Google it maybe. So by combining information from other sources with the observations you made of the bird, you could start making logical conclusions, and that is what inferences are. So you observe your bird, you see what it looks like, what it eats, and then you research to find out what is that bird. So again, an inference is combining what you know with what you have learned from research and to draw logical conclusions from it. So for example, if you Google it and you see a photo of a bird that's similar to the bird that's outside your window, you might infer that that bird is related to the bird in the photo. So if you find a photograph online that maybe doesn't exactly look like that bird, but it's very, very similar, you can infer that those two birds are related. Next, you form a hypothesis, and a hypothesis is a testable explanation of a situation. Now, if you do testing in an experiment and a hypothesis is supported by data from multiple investigations, we would say that that hypothesis is valid and it is accepted. Now, if the hypothesis is not supported, the hypothesis is revised and additional investigations are conducted. And this might be appropriate if you just haven't quite answered your question. So sometimes you can get where you're testing a hypothesis and everything that you do isn't proving it, so you're at least learning what it's not. So that's not always wasted energy to disprove your hypothesis, but that happens sometimes. So next is to do an experiment. So experiments can be different things, but what we're going to talk about specifically are controlled experiments. So what's the difference between a controlled and maybe an uncontrolled experiment? So experiments investigate in a controlled setting to test that hypothesis. And in a controlled experiment, you have to have at least two groups. You'll have your first group, which is a control group. And this is a group that is in the normal, and it doesn't contain the experimental value that you want to test. The second group is an experimental group. This is the test. This is like your test group. I'll move my picture again. So this is your group that's actually exposed to the factor being tested and contains a variable. So what are these variables? We'll go to the next slide for that, and then I'll give you an example. So, sorry, I'm trying to get my photo out of the words. Alright, so during a controlled experiment, it is important to note that only one factor or variable can change at a time. So there's different types of variables that are in consideration during an experiment. So you have an independent variable. This is the tested factor, like the thing you want to experiment. So this thing is what might affect the outcome of an experiment. Then you have a dependent variable, and the results of it depend on the independent. So at least have those keywords. Dependent variable depends on the independent. We also have to consider other variables in an experiment. We call those constants. So since we can only test one factor at a time, there's other variables that might be mixed in there, but we have to keep those the same for our test subjects to make sure it is indeed that independent variable that's affecting the experiment. So let's go through an example. I know we're going through the terms, but let's go through an example. So in this next slide, say I wanted to test to see if cola or orange juice will cause a plant to grow taller. So I have two experimental groups in this one, but it's important to note I have to have a control group in order to compare my results with the other two. So I also have this third group. So I have group A, B, and C is very important in a controlled experiment because I have to have something that's of the normal, and that's what the controlled variable is. So since I normally water a plant with water, that's my control. So I want to see what that does in comparison to watering it with some, weird stuff like cola or orange juice to see what happens. So because the cola is the weird thing, this is my independent variable, my experimental value that I want, or my experimental variable. Or orange juice, it's different. That's experimental. That's the independent variable here. So what's going to be affected by those is the plant growth. Plant growth is depending on what I'm watering it with. So that plant growth is my dependent variable. Here's another example. So say I wanted to test, my hypothesis is that blue color light causes plants to grow more than in white color light. So my control group, I want to put a plant in a normal situation, so I'd put a control group with white light, because that's the normal. Then my experimental group, so trying something new and different, is the plant under the blue light. So my independent variable, in this case, what's being changed is light color. Let me go back. The dependent is plant growth. So my light color is going to affect plant growth. Now remember, controls are things that I have to keep in mind too. So I have to have both plants be the same in all other aspects. So my controls or my constants are going to be the amount of water that I water it with, maybe what time it's watered, or soil type, or the species of plant. Those all have to be exactly the same in order for this to be controlled, because I have to make sure that it is indeed the light that is affecting it. So 4, we have to collect data. So data is information gained from observations during an experiment. So we can have two different types of data we can collect. We can collect quantitative, which quantitative, if you think of that in terms of quantity, has to do with numbers. This is measurements of time, temperature, length, mass, those types of things. Anything having to do with numbers is quantitative data. With qualitative data, we're talking about things that affect the senses. So anything that you can sense, maybe what it looks like, smells like, sound, touch, or taste, anything that has to do with that. So not all scientific inquiry is on controlled experiments, though. Some investigations are just based on observations. We're not actually manipulating any variables. Like when Jane Goodall observed the chimpanzees, she wasn't taking quantitative data necessarily or manipulating variables. She was making just observations on how they behave. So in science, it's also important to note that we use a certain system of measurement, and that is the metric system and the SI system. Now, SI system is the international system, which is like the metric system with only a few small differences, but we use these because they're all based on multiples of 10. So meters, grams, liters, we're going to use those when we're taking scientific measurements. So a metric system uses units with divisions that are powers of 10. SI, or the international system of units, they're commonly used for science because of their consistency throughout the scientific community and for the ease of communication. Now graphs, we will take our data that we gather and maybe put it into tables and graphs. And it's important to note that we'll take the information we get, we'll put it in a table because maybe it's easier to gather the information that way, but we'll format that and put it in a graph. And that's important because now we can see patterns in what the data is telling us. So, if you're looking at this pattern, it's a set pattern, can you make a prediction on how much mass the green anole will have at day 21? Since I don't have a data point there, I can follow the pattern and make a prediction. So if you want to pause the video and think about it, then